Hello, everyone, and welcome to today's webinar, Advancing Immuno-Oncology, Innovative Solutions for Adoptive Cell Therapy. My name is Julia Laschiavo, and I'm from the Prendio BioProcure team, and I'll be your moderator for this session, presented in partnership with PromoCell. We're excited to have you with us as we explore how cutting-edge research tools are helping scientists better understand treatment resistance in cancer, particularly the role of cancer stem cells, and how these insights are advancing the next generation of immunotherapies. Before we get started, a couple of quick housekeeping notes. Today's session is being recorded, and you'll receive a link to the full recording after the event concludes. We'll also have time for Q&A at the end with our speakers, so please feel free as we go through today's presentation. Drop any questions that you have in the Q&A box at the bottom of your screen, and we'll address as many as we can once the talks conclude. Now, I'm very pleased to introduce our speakers for today. First, Dr. Alexander Trompe, Project Manager of PromoCell's Cancer Media Toolbox. Dr. Trompe holds a PhD in cell biology from the University of Lübeck in Germany and brings over a decade of experience as a scientific support specialist with deep expertise in cancer research and cell culture solutions. Joining him is Dr. Courtney Stewart, U.S. HLA Coordinator at PromoCell. Dr. Stewart earned her PhD in biomedical science with a focus on developmental neuroscience and has spent more than 15 years in scientific education and research. She's passionate about helping researchers apply advanced cell culture and HLA-typed models to their work. So with that, I'll remind you again to please keep any questions coming that you might have in the Q&A box, and I'll hand things over to Dr. Trompe and Dr. Stewart to begin today's presentation. Thank you.
Yeah, thank you very much for the introduction, and also welcome from my side to today's webinar. Yeah, as you know from the title, today's focus will be on adoptive cell therapies, one section of immuno-oncology. You could say one of the most dynamic and promising fields in cancer research. But let's start first by looking at the current landscape of immuno-oncology in general. There has been quite a paradigm shift in cancer treatment, moving from traditional chemotherapy towards strategies that harness the body's own immune system. In the 1950s, it started with microbial therapies. Then by the 1980s, we saw the emergence of cytokines as the main focus, but as well as monoclonal antibodies and tumor-infiltrating lymphocyte therapies. The 2010s brought checkpoint inhibitors as a major breakthrough, and also CAR T cells alongside bispecific antibodies and more refined cytokine approaches, they emerged as well at that time. Now in the 2020s, the field has expanded even further and is more diverse than ever in its approaches. There are nowadays, besides the checkpoint inhibitors, also antibody-drug conjugates and vaccines in the mix, while researchers continue to refine earlier strategies. So we've really come a long way in the field. But as Jeremy Levin said from Ovid Therapeutics, the first wave of immuno-oncology has passed us by, and the field is now at a critical point. It's lost a little bit of momentum and is looking for a new breakthrough. So in the next part, I'm going to look at three of the prominent strategies that are currently in the focus of immuno-oncology, which are the immune checkpoint inhibitors, antibody-based therapies, and adoptive cell therapies. So the first one, immune checkpoint inhibitors. They are drugs that work by blocking immune breaks like PD-1, PD-L1, and CTLA-4. Under normal conditions, these checkpoints help prevent overactivation of the immune system, but cancer cells exploit them to escape immune detection. So by inhibiting these checkpoints, we can restore T cell activity and enable the immune system to recognize and detect tumors. So far, around 14 checkpoint inhibitors have been approved by the FDA, and they have truly transformed treatment for several cancers like leukemia and lymphoma. However, response rates have plateaued, and both primary and acquired resistance remain major hurdles. Other challenges include barriers to reaching the tumor microenvironment or causing immune-related adverse events and limitations in the availability of biomarkers. As a result, the approach is shifting. Preclinical data suggest that success depends not just on what we combine, but also on how and when we do it. So timing, sequence, and immune context all matter. So there's now growing interest in next generation checkpoint targets beyond the usual suspects. And yeah, as mentioned, there's now the shift towards combination therapies. For example, pairing these checkpoint inhibitors with other agents. One of them, for example, are bispecific antibodies. They are versatile constructs that bind both a tumor-associated antigen and, for example, an immune cell. They can directly lyse tumor cells or selectively engage immune cells in the tumor microenvironment. Other antibody-based strategies are the antibody-drug conjugates, ADCs, and so-called ISACs, the immune-stimulating antibody conjugates. Especially ADCs have become one of the fastest growing therapeutic classes in oncology. They combine the targeting precision of monoclonal antibodies with the potency of cytotoxic drugs. So they're offering this targeted cytotoxicity, but with a reduced systemic toxicity if, for example, you compare it to traditional chemotherapy. What's also promising is their immune modulation potential. These ISACs, for example, they can deliver immune activating payloads like TLR or STING agonists directly into the TME. And there's growing evidence that ADCs can reprogram the TME and enhance anti-tumor immunity, especially when used in combination with checkpoint inhibitors. These therapies still have the challenges. Despite their precision, off-target toxicity remains a concern, and manufacturing is complex, costly. There's like high demands on consistency and stability when it comes to the antibodies, the linkers, and the payloads. Payloads also, there's like a limited diversity so far. Many cytotoxins that are used for that are, they would be too toxic or they turn out to be unstable for clinical use. And then, of course, there are the pharmacokinetic challenges that they make it difficult to predict distribution and clearance. So dosing strategies can also be a challenge. So while this approach holds quite a great promise, especially in combination settings, it still requires further optimization. Now, looking at adoptive cell therapy, so the approach that leverages the immune system by isolating and then engineering its components to fight cancer. The concept is to take immune cells from the patient or a healthy donor. So using either autologous or an allogenic approach. And then to modify and or expand these cells in the lab and then infuse them in patients to enhance anti-tumor activity. Tumor-infiltrating lymphocytes, TILs, for example, are collected from resected tumors, so tumors that can be reached surgically, and then expanded in vitro and reinfused. They recognize tumor antigens in an MHC-restricted manner and have shown clinical benefit, including an FDA-approved product for metastatic melanoma. Besides the TILs, there are the T-cell receptor, the TCR-engineered cells, and the chimeric antigen receptor, the CAR T cells. And they are currently dominating the field. Both start with T cells, which are genetically modified and expanded before infusion. Difference is that the TCR T cells use natural TCRs to recognize antigens presented by MHC. And CAR T cells replace the receptor with an antibody-derived construct that then binds tumor-associated antigens directly on the cell surface. And as time progressed, more and more immune cells are also entering the stage. For example, NK cells for innate tumor killing, CAR macrophages are used to boost phagocytosis, And also B cells are engineered to enhance antigen presentation or immune modulation. And most likely, as time will pass, more cell types will appear on this list. So CAR T cell, you can really say it has revolutionized the treatment of hematologic cancers. And it's been delivering remarkable clinical outcomes in diseases like leukemia and lymphoma. However, progress in solid tumors has been slower, although there are promising developments now, particularly with TIL therapy, for example, in this melanoma mentioned. And recent advancements also include the design and development of these multi-targeted and cytokine-enhanced so-called armored CAR T cells that are designed so that they have a better persistence. And yeah, as mentioned, new cell types are being tested. For example, lately, the invariant natural killer T cells, which are showing strong potential in preclinical models for solid tumors. These innovations reflect the broader trend that you can see towards engineering immune cells for greater potency and also durability and versatility. Together, the approaches are very promising, though they are also still facing challenges. One major hurdle is the antigenic heterogeneity that you see, so that tumors often display a diverse set of antigens, and that is making it difficult to identify a single reliable target. And even if you find suitable targets, they're often expressed at low levels also on healthy tissues, which then, of course, is leading to on-target off-tumor toxicity, where engineered immune cells then attack normal cells. Then there's the issue of cell trafficking. Reaching the tumor microenvironment and infiltrating it with the modified cells is not straightforward, as there are physical and biological barriers that limit access. And then, even if the immune cells are reaching the TME, they face an immunosuppressive environment that can deactivate or exhaust them. Now, many of these challenges might be amplified, if not caused, by a certain subpopulation of cancer cells, which are the cancer stem cells. So just to recap what we're talking about, these cancer stem cells are often also called cancer stem-like cells, or in short, CSCs. They are a rare subpopulation within tumors, typically accounting for only 0.01 to maybe maximum of 2% of the total tumor mass. They share key properties with normal stem cells, such as self-renewal and differentiation. And they can originate from normal stem cells or progenitor cells that acquire stem-like traits through mutations. Because they can differentiate into various cell types within the tumor, they maintain the tumor's heterogeneity, and they also drive its continuous growth. They're also capable of initiating tumor formation. So since they're also playing a role in metastasis by migrating to distant organs, they can then start new tumors there. So as you can see, many significant hurdles I mentioned earlier may be directly or indirectly linked to cancer stem cells. So first, they contribute to immune evasion because CSCs are known to suppress immune responses and help this immune suppressive microenvironment that protects them from attacks. They do that by secreting inhibitory cytokines and also by expressing these immune checkpoint ligands, which then reduce T cell activation and persistence. Also, they're also a tricky target because they have a low MHC expression, so it's harder for immune cells to recognize them. Second, they contribute to the heterogeneity and also plasticity because CSCs can shift between a stem-like and a non-stem-like state, and that complicates targeting. Third, they contribute to resistance and relapse. CSCs are pretty good at surviving treatments. They can go in a quiescent state, avoid immune detection and this therapy-induced stress. They can do that by having this enhanced DNA damage repair and also higher expression of these drug efflux pumps, so that helps them to have a higher resistance to these treatments. And then post-treatment, they can re-enter the cell cycle and just repopulate the tumor. Yeah, and finally, the issue of on-target off-tumor toxicity. CSC markers are frequently shared with normal stem cells, so targeting them risks also, again, healthy tissue. So often, if potential targets are identified, they frequently overlap with normal stem cells. And that makes it then difficult to design therapies that are both effective and safe. So this lack of truly tumor-specific antigen is like an ongoing challenge. And together, these hurdles show why CSC should be a critical focus in next-generation immunotherapy. As, for example, Chu et al. In 2024 noted, the presence of cancer cells can be an essential factor that leads to failure of cancer treatments. Because if cancer stem cells are not targeted, the risk of relapse remains high. Because conventional therapies often fail against CSCs because of their therapy resistance. So they are often allowing the CSCs to survive treatment and then repopulate the tumor. So by targeting CSCs alongside the differentiated cancer cells, we can then eliminate tumors more completely, reduce recurrence, and achieve better patient outcomes. So including CSCs in patient-derived cultures is especially critical for drug discovery. If you are having them present in models, that helps creating more accurate models of the team E, and that in turn then enables development of therapies that effectively target CSCs and improve the long-term success of treatments. So, of course, there's now the question, how can we address these challenges? And there are 3 hurdles to developing robust adoptive cell therapy that we're going to talk about now. One is how do I isolate these cancer stem cells? So how can I establish like a good primary cancer culture? That's foundational, because without a reliable way to enrich CSCs from patient samples, we can't create a model that truly reflects the tumor biology. Then second, once we have them, how can we co-culture the CSCs with the engineered immune cells in 2D or 3D? Because that step is then critical to study or test immune tumor interactions. And third, finally, how do you get a cell culture control panel of HLA-typed cells to test for on-target off-tumor effects? That's important for the safety. And this testing against normal cells will then help to detect any unintended targeting and assess toxicity before you move into a clinical setting. So talking about isolation of CSCs, there are some barriers and some gates that we would like to cross. Ideally, you want a method that doesn't require any specialized equipment, but just easy handling. You want something that preserves the tumor heterogeneity and without introducing any bias. Often you have the problem that fibroblasts overgrow your culture, so you want to prevent that, but support then the long-term cultivation of your CSCs. Because all that will then help that you can make good use of a biopsy that you have and really start a patient-derived culture. So to achieve this first point, we are offering the primary cancer culture system, or in short, the PCCS. This method that I'm going to present now for isolating CSCs from patient tumors or cell lines relies on the functionality of this cell culture medium that we at Promocell have developed. It's called the primary cancer culture system. It's a defined animal component-free and complete cell culture system, so you don't need to add any other additives. The key feature is that it isolates and enriches for CSCs from a variety of starting materials, such as primary tumor tissues, cell lines, or PDX biopsies. It's pretty much a universal medium. And it functions by actively depleting benign cells so that only CSCs, due to the unique metabolism, survive. And then the CSCs can thrive in this medium, while benign stroma cells like fibroblasts, for example, are starved and killed. So I'd like to go with you now through the workflow and also show some data of an isolation we generated in-house. Here we have isolated cancer cells from biopsies of liver metastasis. The original tumor was a gastrointestinal stromal tumor. The workflow starts with the homogenization and enzymatic digestion of the biopsy, the tumor tissue that you have, to create this heterogeneous single cell mix of different cell types, such as the CSCs that are hopefully present in this case, for example, and differentiated cancer cells, normal cells like CAS, for example. Then this single cell suspension is plated on tissue culture vessels that are pre-coated with an NCCD reagent, and then allowed just with fresh medium added around day 6. After 4 days of isolation, here's a picture from that point. You will see a lot of floating multicellular aggregate dead cells. At that stage, as mentioned, CSCs make only like 98%, 2% of your tumor mass, so 98% are being killed off at that stage. So it looks terrible, but that's exactly what's supposed to happen. The medium only supports the CSCs in the long term, while all the other cells are starved out. So after the initial plating, then the selection process continues for 10 to 14 days. And depending on the tumor, depending on the biopsies, the cells might switch from suspension growth to adherent growth or the other way. It's what we've seen so far. It's something that the tumor decides. The tumor decides the growth pattern. The culture then continues by simply cultivating it in the PCCS, so no sorting or any other hands-on steps are needed. This image here shows the culture after 14 days after the initial plating and after one passage. And you can see the culture has cleared up and the primary GIST cells appeared here as cell clusters. We then continued that culture, and after 2 to 3 weeks, the CSCs started to grow. We could expand them by regular media changes and passaging in the PCCS. And here the picture after day 17, you can see the GIST cells started to proliferate and exhibited this spindle-shaped morphology. That's what we would have expected also, since that's the typical morphology of stromal tumor-derived cells. Then we could expand the cells further. We have passaged this culture for 49 days, and at the end, we could really say that we established a stable primary cancer cell culture that we could keep for long-term cultivation, and we were even able to freeze down some aliquots to form like a stock for later studies. Of course, we ask ourselves, are these really cancer stem-like cells? And yes, we think so, because they have a very low proliferation rate that's typical for stem-like cells. The population doubling time averaged around 200 hours, which is characteristic of slow-growing cancer stem cells. So for comparison, normal fibroblasts, they have a doubling time of around 24 to 55 hours. Second, we did some FACS analysis, where we stained the cells with antibodies using against the stem cell markers OCT4 and SOX2. And we could see that 97% of the isolated cells were double positive for these stem cell markers. Also, we then tested the cells for their ability to initiate tumors. To check that, biopsies were taken from PDX mice and cells isolated using our PCCS. Then from these primary cancer cell cultures, cells were taken and re-engrafted into mice, and there they indeed initiated new tumors. So that confirmed also their tumor-inducing capabilities in vivo. And all this suggests that the cells isolated with our PCS are really a pure population of cancer cells with distinct stem cell properties and self-renewal potential. And all that, as you see, without the use of markers and thereby without introducing any bias. And this is important because by preserving the malignancy and heterogeneity, we can ensure that the research is more relevant and reflective of in vivo conditions, which means that the original cancer cell subpopulations need to be maintained. Thus, it's important to eliminate marker bias and avoid conventional media where the growth of CSCs are not supported and that the CSCs are lost in the long term. So our PCS functions across various tumor types as this metabolism, the special metabolism of CSCs is shared among tissue types. And then you don't need any marker-based sorting. Here, just quickly, is another example of how you can also use the PCCS with a few tweaks of the protocol and the addition of cytokines. It can be used here, it's like another experiment that we did where it was used for the isolation of TAMs, of tumor-associated macrophages from primary tissue samples of a solid tumor. So here we could then isolate the TAMs as non-proliferating adherent cells, and we could culture them for 2 to 3 weeks. It's also an application note you can find on our website. It's called Isolation of Tumor-Associated Macrophages from Fresh Tumor Tissue. Just let us know later if you would like the link. We're happy to send it to you. So as you can see, our PCCS doesn't require anything special. It's just easy cell culture. It preserves your tumor heterogeneity without introducing any marker bias. And you can prevent fibroblasts overgrowing the culture because it supports the long-term cultivation of CSCs. So that, yeah, you can make good use of your biopsy and really create like a stock for future experiments. So now that we have our CSCs isolated and established like a permanent culture, how can we co-culture them with our engineered immune cells? Here we can offer 2 solutions as part of our cancer media toolbox. One for 2D applications, the cancer cell medium XF, and for 3D applications, the 3D tumor cell medium XF. It's no problem at all to switch back and forth between our cancer cell culture media because the PCCS, the cancer cell medium, and the 3D tumor cell medium, they're all compatible with each other. So you have the full flexibility to run assays in 2D or 3D. For example, let's imagine you need to test newly developed CAR T cells for efficacy against a patient-derived thyroid model. You can isolate the CSCs using the PCCS and then generate 3D tumor spheres by switching to our 3D tumor sphere medium. The immune cells, they can survive in our cancer media as they are for short experiments. And if you add the necessary cytokines, immune cells will even proliferate and survive longer. So to go a bit more into 3D cancer cell culture, here you have the similar goals. You want to create your own patient-derived reculture, establish uniform, robust tumor spheres, and then scale up the model. The barriers that you have there are that you often need different media for different cancer cell lines, different tumors. There's a danger of losing the original traits and complex protocols and reproducing results. Now imagine if you can use a single standardized protocol instead to maintain the crucial CSCs and have a more robust protocol. For this, we're offering our 3D tumors via Medium XF. It's a serum and xeno-free medium that supports diverse starting materials also, like these primary tumor tissues, the cell lines, and the PDX cultures. It enables the formation of homogeneous 3D spheres through serial passaging by enriching and then maintaining the CSCs that you have alongside proliferating differentiated cancer cells. So that's consistent and the key for long-term 3D culture models. We've tested a lot of various cell lines for multiple tumor types, including melanoma, colon, lung, breast, prostate, liver, connective tissue, and brain. And you can see here we could passage them up to 10, 11 passages. This shows you can use the same medium across different research projects and you do not have to switch between different media. If you think about drug development, where reproducibility and consistency is key, this xeno and serum-free formulation is really an advantage. Also, the 3D tumors for medium has proven to be a solid foundation for advanced 3D models, including bioprinting, generation of organoids, and hanging drop methods. And due to the medium's versatility, it's proven to be complex, suitable for complex tumor modeling approaches that better replicate in vivo conditions, and that gives the opportunity to establish such models without an extracellular matrix. Now, developing 3D tumor sphere models from biopsies and cancer cell line aims, of course, to establish a model for screening and assay panels so that you can identify and test drug candidates. And such more complex models need the presence, of course, also of non-cancer cells, such as immune cells or fibroblasts, and maybe also ECM components. For these more complex models, we can offer you the cancer cell line medium. It's a serum-free, xeno-free medium without any undefined components like FCS, extracts, or hydrolysates. And that ensures like a minimal lot-to-lot variability and provides a well-defined culture environment, which is ideal if you rely on reproducible conditions. That medium also supports both CSCs and more differentiated cancer cells, but at the same time, it's also allowing non-cancer cells, such as fibroblasts, to grow, and that makes it especially suitable for co-culture applications. And also you can combine it then with attachment matrices, with collagen, matrigel, other ECM components, if you want to mimic tissue architecture or tumor stiffness. So here's one example where we use the medium. It's also from an application note that we have called cancer organoid models. It's a method for 3D organoid culture. In this case, we used dermal fibroblasts and MCF7 cells. We cultivated them in the cancer cell line medium, first like expand them separately, and then harvesting them and seeding them together for organoid formation. Organoids then formed spontaneously within 24 to 48 hours of incubation, and then tightly packed organoids formed within 4 to 5 days. Here are the exemplary results after initiation of the organoid culture in the collagen gel. In A, you can see that only organoids containing MCF7 cells and fibroblasts, they showed the characteristic sprouting by the fibroblast, here marked with the red arrows after the organoids were embedded in the collagen gel. If you want to learn more about the setup, let us know, and then of course we can also provide you to the link to that app note. As you can see with our cancer cell culture media, you can standardize your protocols for different tumor types, you preserve the original traits because the CSCs are supported, and you can then establish these long-term cultures so that you have a good 3D, patient-derived 3D model that you can then scale up. So these are the three media making up our cancer media toolbox. And by choosing one of the media, you can control which additional types of cancer or non-cancer cells are supported, or vice versa, which cell types will not grow in the medium. Depending on the medium and the cancer type, you can then do 2D or 3D cultures. So you got the PCCS, highly selective for the cancer stem cells. For 3D application, the 3D tumors medium without ECM, and the cancer cell line medium for co-cultivation and with a matrix also for 3D applications. And all three media, they support and maintain the cancer stem cells so that independent of their origin, you can cultivate them. They're really universally applicable. So these are our three cancer cell culture media that help you to isolate and then cultivate the target cells and then co-cultivate them with the engineered immune cells. Now, to address the third hurdle, I will hand over to my colleague Courtney to talk about HLA-typed primary cells.
Thanks, Alex. Hi, everyone. Again, welcome. Thank you for attending. We are going to now talk about the third hurdle and what that means when you are in a preclinical or clinical setting. When you're developing these cancer therapies, key things to consider would be the efficacy of that therapy, the safety, and the ability to further personalize it. So one way to do that would be to use HLA-type cells, specifically for on-target and off-target toxicity, as well as a few other things that we'll go over in future slides. So to begin, just a little bit of background on HLA. HLA is the human leukocyte antigen system, or also known as the major histocompatibility complex, and it's one of the most complex and variable regions of the human genome. These are cell surface proteins that regulate the immune system in many ways, and they're extremely diverse. So you have over 2,000 HLA class 1 alleles and over 600 HLA class 2. HLA research has transformed our understanding on how these immune cells interact with one another in our body and how the immune system can prevent disease. But a few things to consider here with the class 1 before we move on is that these particular, sorry, we're losing control of the mouse here. These particular cells are expressed on all nucleated cells and platelets and present the antigen from inside the cell, mainly presented to T cells, whereas class 2 are specific for APC and present antigens and peptides from outside the cell in a non-pathogenic self-antigen or pathogenic classification, and they're presented to T cells, B cells, and NK cells. These molecules also help the body to distinguish your self or foreign substances, such as pathogens, and they're important for transplant or non-self roles in response to disease susceptibility and transplantation outcomes. They've also become very important in understanding the role of the immune system in autoimmune diseases and the defense against other infections and further personalizing medicine. But specifically for cancer, they're driving a lot of breakthroughs in novel immunotherapies, considering that tumor-reactive T cells can recognize cancer cells through these HLA molecules and also help determine the effectiveness of therapies like ACT, which depend largely on that compatibility. HLA also will help identify the likelihood that the patient will respond to immunotherapies, and they're further used to test new cancer treatments. So if you're in a preclinical setting, one of the key things you want to do is minimize any off-target effects that that particular therapy might have. So here at PromoCell, we do offer a wide selection of high-quality HLA-typed primary cells from a variety of human tissues. So this could give you many tools to evaluate these therapeutic candidates and detect any cross-reactivity that you may not want early in your development. In short, We will help you move faster, test smarter, and develop new therapies more efficiently, saving you time and resources. So just to summarize, before I hand it back to Alex, again, some key things to consider when you're in the preclinical or clinical setting would be therapeutic efficacy, safety, the personalization of therapies, and potential tissue rejection in the case of transplants. You can minimize off-target toxicity effects after conducting these assays, as well as determine the on-target effect, how efficacy will work in your system. In addition to immune compatibility, which can further determine whether or not a patient will respond to that therapy, or even use HLAs as biomarkers to see their likelihood of responding to those therapies. Again, at PromoCell, we do offer a wide variety of HLA-typed cells, which will allow you to further develop an easy panel that is assay ready, with minimizing the time and financial burden on yourself, since you do not need to do the HLA typing on your own.
Yeah, thank you, Courtney. So this is how we can help you address the challenges in the development of adoptive cell therapies. The PCCS allows you the isolation of cancer stem cells without fibroblasts overgrowing your culture and without introducing any bias from any type of tumor tissue. And yeah, our 3D tumor sphere medium serves as well as one medium for all 3D cell cultures, independent, for example, of what cell line you're using. And it allows you to establish tumor sphere cultures for up to 10 passages, so really long term. And the cancer cell line medium is your go-to solution for co-culture experiments of cancer and non-cancer cells under highly standardized conditions. And as Courtney just described, our portfolio of HLA-typed human primary cells provides you with an assay-ready cell culture control panel, and there's no need to do any HLA typing on your cell, on your end. So that's it from our side for now. We're going to have a look now if there are any questions in the chat.
Yes, absolutely. Thank you so much to Dr. Trempe and Dr. Stewart for a great presentation. I do have some questions for you. If folks have additional questions, please feel free to keep them coming. You can use the Q&A box at the bottom of your screen, but I will get things started with the first question. Someone is interested in, they're investigating extracellular vesicles, including exosomes from cancer cells, and wants to know if your cancer media is EV depleted.
Not that we are kind of mixing the media and then depleting it for EVs. They are automatically low in EVs since we are not using any serums, any hydrolysates, for example. And especially the cancer cell line medium, it's been tested by some customer, and they found that it's like I think less than 1% of what you would even find in an EV depleted medium. So it's virtually EV free, and you can really use it then, for example, to cultivate your cells, produce EVs and harvest them.
Okay, great. Thank you. The next question I have, someone is having trouble establishing a primary cancer cell culture from some biopsies with their media that they're currently using. And they're wondering if you think the primary cancer cell culture system would work with those hopeless samples.
Yeah, hopeless. It's, well, okay. It's I have to say the PCTS is not a magic potion, meaning it won't like generate cancer stem cells. And there are some factors that can kind of influence if the biopsy that you get is contains CSCs or not. It's like if it's like a hopeless biopsy that doesn't have any cancer stem-like cells, or if it does have them. I mean, it's a little bit of a game of luck. If the surgeon excised and sent you the portion that has the CC niche, that's of course one factor. And if you have a chance to ask them, it's also good to know if the patient has just recently undergone any chemo or radiotherapy. If that's the case, we also found that chances of isolating cancer stem-like cells really drops. Plus, how long did it take for the biopsy to get to your lab? I think best results we had were like with tissues up to 6 hours, but it also worked with like tissue samples that were like 24 hours old or maybe even a bit later. So yeah, it takes two to tango. There is a medium, but also there's a biopsy, and I think the biopsy also has a crucial effect on how well it works. But we've definitely seen that you have a way better chance of isolating the cancer stem cells without PCCS, especially since a lot of people that we also talk to, they have often the issue that they start a culture and then in the end it's just fibroblasts. And that's something that we can really prevent with our PCCS.
Okay, great. This next question also touches on PCCS. So when isolating CSC from patient-derived cells, you are using PCCS. But when performing a medium change, do you still use this medium or the conventional one?
You would stick with the PCCS. The PCCS is like the selective one that will keep like the culture that only like the cancer stem-like cells are supported. If you would switch to yeah, a standard medium, for example, if you do that early, there's still a chance that maybe some fibroblast survived and then they might expand. Although the longer you have it in the PCCS, the less likely that is. Of course, also you would have to be aware that if you switch from the PCCS to like a normal medium, the stem cells, they will start to differentiate. You will have like a mix of stem-like cells, but also differentiated cancer cells. So we always recommend that you keep like a backup culture just in the PCCS.
All right, great. Thank you. So this next question, excuse me, touches on HLA. So Courtney, I think this is for you. What resolution is used for HLA typing?
At PromoCell, we use a 4-digit resolution system that is determined after detecting which allele is present. So we use RSSO or reverse sequence specific oligonucleotide probes that are then bound within our system and ran on a Luminex multiplex bead analysis to determine what DNA is encoding that particular genome.
Okay, great. Thank you so much. Next one I think is for Alexander. Someone is interested in growing organoids from lung cancer cells in blood cells and wants to know which medium you would recommend.
Um, if it's just like the cancer cells, I would say the 3D tumor sphere medium. But if the question means like if you want to grow them together, do some co-culture. I assume blood cells means immune cells. You can also do that in the 3D tumor sphere medium. If you add, as mentioned, like cytokines, they will also survive and proliferate there, also long term. If you really want to do then also co-culture with other cells, then I would maybe try the cancer cell line medium because it's the least selective, and that will also really allow non-cancer cells to grow.
Okay, thank you. Now you mentioned the 3D tumor sphere medium. There's another question wondering how it works. Specifically, how does it keep the cells in suspension?
Yeah, as mentioned, it's serum-free medium, so it naturally doesn't have any attachment factors that the cells would need to attach. So if you then also we in the protocol you can see that we recommend using it then also with low attachment plates, and that's just like not giving the cells any chance to attach and forces them in suspension, and that way they stay in suspension and you don't need an ECM.
Okay, great. Another HLA question for Courtney. How does PromoCell determine the HLA type of its cells?
That's the RSSO, the reverse single sequence oligonucleotide probe, ran again on Luminex, just to determine what DNA is encoded at that point within that sample.
Okay, excellent. I think we have time for just a couple more questions. Again, if there are any last final questions that you'd like answered live, last call for those. Last question I have is, which medium is recommended for the co-cultivation of cancer cells with CAFs or normal cells?
Yeah, the cancer cell line medium, because it's not selective, or it wouldn't eliminate like the cancer-associated fibroblasts, for example. That medium I would pick.
All right, got it. Okay, so that is all the questions we had today. Alexandra, I believe you have a couple more slides just to tell the audience here. And as a reminder, everyone will receive the link to this recording. And our speakers have generously offered, if you have any questions following this session, that you are more than welcome to reach out to them.
Yeah, here are our contact details. You can ask me any question about the cancer media, any questions about the HLA type cells. Courtney is your contact person. Feel free to reach out to us by phone or by email, and we're happy to get back to you. And also an invitation, if you happen to be at the SITC annual meeting, it's like November 5th to 9th in National Harbor. We are there with a booth. It's booth number 930. And yeah, we'd be happy to meet you in person and then discuss your applications in detail over a cup of coffee. Yeah, and that's it from our side. Yeah, so from my side, thanks a lot for attending the webinar. And yeah, let us know if you've got any other questions.
All right, thank you so much. Thank you to Dr. Trompe and Dr. Stewart for your expertise and your time today. Again, everyone will be receiving the link to this recording, and please be in touch with any questions. And with that, have a great rest of the day. Thank you.
Thank you.